Our lecture today, this is lecture number 14, and again, we're still in the area of learning, and we've taken a look at classical conditioning and taken a look at operant conditioning and tried to understand some of the fundamentals of learning. And what I want to do now is I want to compare these two different types of learning. And that comparison becomes very important for some of the major implications of these two different types of theories, learning theories. And the implications are in terms of application, because we're going to talk a little bit about what's called biofeedback, which is a consequence of understanding these two different types of learning. So let's begin this analysis by comparing these two types of learning. And again, first of all, in classical conditioning, if you just take a look at this little portrayal right here, classical conditioning, we're learning to associate an involuntary response and a stimulus. So again, recall Pavlov's dog that associated a bell with the presentation of food. In the case of operant conditioning, we're associating a voluntary behavior and a consequence. And again, the voluntary behavior that was being studied by B.F. Skinner was that of pressing a bar, a rat pressing a bar in order to obtain food. So let's again make sure that we understand the distinction between these two types of learning, because that distinction, once again, is really going to be very, very important as we go along to understand the implications of this work for one of the major developments in psychology over the last 40 or 50 years, which again has to do with this whole topic of what we call biofeedback. So in operant conditioning, we're dealing with voluntary behaviors. You know, case of an animal that's in a Skinner box, there's a wide array of these different voluntary behaviors that an animal can exhibit. And these responses are responses that we call emitted responses. Those responses, we're going to select out one of them and we're going to try to encourage it. And the reason, the way in which we go about encouraging it is by way of reward, by way of reinforcement. So the organism, again, if you take a look at the original experiments by Skinner, the organism is learning how to operate on its environment. It knows what behaviors, what responses, what voluntary responses are going to be reinforced and which ones are not going to be reinforced. And again, those that are reinforced are ones that are now going to be strengthened. One of the important aspects of operant conditioning and operant learning is that this is a behavior that really requires type of learning that really requires the utilization of higher brain areas. Again, one of the things that we know is that the cortex of the brain, you know, the smart part of the brain, is really very much involved in this kind of behavior, this operant conditioning. Now, classical conditioning, on the other hand, again, a more fundamental, a simpler type of learning. We know that what we're really looking at are involuntary responses. These are automatic responses. These are responses that are elicited or drawn out of the organism. Again, when you take a look at the relationship between food and salivation, you know, placing food upon a dog's mouth automatically produces this salivary response. So again, in the case of classical conditioning, we're really dealing with different types of responses entirely. We're dealing with involuntary responses. We don't really need reinforcement. It's contiguity. It's the closest in time of two events. That's really crucial for classical conditioning. So what is happening here? Well, what is happening is, again, just the reverse of what we see in the case of operant conditioning. The environment is operating on the organism, again, to draw these behaviors, these involuntary responses out of the organism. And we know that lower brain areas are involved in this behavior. There have been, some fascinating studies that have been done, for example, with a technique which is called spreading depression, in which scientists can temporarily deactivate the smart part of the brain, the cerebral cortex, by way of a chemical treatment, a reversible chemical treatment. And then what they can do, even though the cortex has more or less been taken out of the equation, they can still teach an animal a classically conditioned response. So those higher brain areas really are not needed for classical conditioning. So again, some important distinctions between these two different types of learning. So here's a research question, and it's one that probably won't make a lot of sense to you right now, but I think will make sense to you as we go along. And that question is this: is it possible to operantly condition an involuntary response? Again, we have a wide array of involuntary responses, things like blood pressure, for example, heart rate. These are all involuntary responses. Would it be possible to modify those internal physiological responses by way of operant conditioning? Now, again, that's not something that probably makes too much sense to you right now, but hopefully it will make sense to you as we go along when we talk about some of the research that really has led to this very important technique in the field of psychology, which is called biofeedback. So let's talk now about this very interesting experiment performed by James Olds, who you see there, and his then student at the time, Peter Milner, in which they were exploring this area of the brain that you see right here, which is called the reticular activating system, which at the time a psychologist believed was involved in the arousal process. And what they speculated was that an animal that's slightly aroused is probably an animal that learns better because it's more aware of its environment and is more attentive to things going on in its environment. And Olds and Milner thought that if they took an animal, a rat, let's say, and placed it into a difficult maze, a maze that they were experiencing for the first time, that if they were able to stimulate that arousal system in that rat would probably learn that difficult task more rapidly than a rat that was not stimulated in the reticular activating system. This was an interesting hypothesis and indeed one that they wanted to explore. But they had also been reading about some other research at the time that indicated that there were certain parts of the brain that are actually ones which, when stimulated, have painful negative effects. And they thought that they wanted to make sure that this was something that was not happening, because that could completely alter the outcome of their studies and complicate their studies. So what they did was this: they took experimental rats and they placed them into a box like what you see here. This is a view of this chamber that they used that was, four-sided. This is a view from the top, and it was divided into four different quadrants simply by painting a line here horizontally and another line here vertically. And so now we have four quadrants. Here's the first quadrant, the second quadrant, the third quadrant, the fourth quadrant. And what they did was they used a large number of rats that had an electrode placed down into the reticular activating system. They would place them in this box one at a time, and they would simply allow them to wander around to wherever they wanted to go. And what they did was they allowed the animal to wander around a little bit, but whenever, for example, with the first rat, whenever the rat went into this third quadrant right here, into this corner of the third quadrant, what they would do is they would provide a little bit of stimulation to the brain, okay, to the reticular activating system. And then what they would do is they would allow the animal simply to explore and continue to explore and examine, how much time they would spend in each one of these quadrants. So again, whenever it goes here into this corner, it's going to get a little bit of stimulation to the brain in the reticular activating system. What Olds and Milner found was that the animals, even though they received some stimulation here, they explored all parts of this chamber, spent an equal amount of time in here and here and here and here, which indicated to them that this stimulation was not something that was having a negative or aversive effect, because they thought that the animal would probably avoid this area if that stimulation to their brain was producing a painful negative effect. They would not want to go back to a place where they had received that, but that was not the case. They were spending equal amount of time in each one of these areas of the cage. Well, they did this with animal after animal after animal after animal, and they continuously found, the same kind of an effect. They were spending equal amounts of time in each one of the chambers. So some of the animals, for example, got stimulation here. Some of them got stimulation here. Some got stimulation here. Some got stimulation here. It didn't matter, right? They would spend equal amounts of time in each one of these corners or quadrants of the cage. But then there was one animal, and this is just such a wonderful case, really, of serendipity. There was one animal where the animal was in the fourth quadrant, and that's where it was being stimulated. And when the animal was stimulated, its ears kind of perked up, and the vibrissae, the whiskers, if you will, would start whisking very, very fast. And the animal retreated, but then came back right away. And again, it got another stimulation. And the animal would explore around, then come back again really rapidly, as if it liked it. It was as if they found it to be pleasurable. And indeed, this one animal, which was so different from every other animal that they included, they were scratching their heads, Olds and Milner. And what they did was they explored the brain of this animal to see if the electrode had definitely gone into the reticular activating system. And what they found was that instead of it going into the reticular activating system, they had made a mistake, and it instead went into a part of the brain called the medial forebrain bundle. That we now know is the reward center of the brain, the reinforcement center of the brain. Let's take a look now at how Olds and Milner now pursued this very interesting finding. They stopped all of their research now and began to take a look at what would happen if you took a rat, placed an electrode down into its brain, into the reticular activating system, and then teach the animal to press the bar to get rewarding brain stimulation, which is easy to do. And indeed, what they found was that the animal would spend all of its time just sitting there, pressing this bar as rapidly as they could, in some cases several thousand times an hour. What they also found was that the animals would prefer to do this. They would prefer to press the bar to get this rewarding stimulation as opposed to eating food that they really liked, or in the case of a male rat, engaging in sexual behavior with a female rat. They preferred to be spending all of their time simply pressing this bar as rapidly as they possibly could. So this proved to be one of the most important findings in the field of psychology, because we now know that the part of the brain that's involved in how we experience reward, and we know now is involved in all different types of addiction, is this network here, this pathway here of dopamine neurons. And one of the key areas is this area that we see right here, which is called the nucleus accumbens. Here's our medial forebrain bundle that you see right here. And indeed, we know that this is the reward pathway of the brain, and has helped us enormously to understand many, many different types of addiction. Now, this study is a pivotal one. It's a pivotal one for understanding a study that was to come a few years later. And this was a piece of research that was conducted by another pioneer in the field of psychology by the name of Neil Miller. Here's Neil Miller right here at Rockefeller University for a number of years. And what Neil Miller was able to do was to shape, that is, modify, reinforce, okay, heart rate. Reinforce it in such a way to increase heart rate, or reinforce it in such a way to lower heart rate. And the experiment itself is a very interesting one, and I want to make sure that everybody understands it. So Miller's research consists of the following. Rats were immobilized by way of administering a drug to them, which is called curare. Here's what curare does. If I had a rat right now and I had administered curare to it, the rat would not be able to walk around, but its eyes would be open, and it could hear, it could smell, but it simply wouldn't be able to walk around. So the animal now, being administered this curare, this drug, cannot exhibit any voluntary responses. Okay. So what Miller is going to do is this. He's going to implant an electrode into the brain of the animal, into the medial forebrain bundle, the reward area, right? And he's going to monitor the heart rate of the animal. And every time he sees that the heart rate, heart rate is something that fluctuates, obviously. Sometimes it goes down, sometimes it goes up. But whenever the heart rate of the animal slightly increased, what Miller would do is to give the animal some rewarding brain stimulation, right? Maybe about a half-second long, a brief pulse of electrical activity to the brain. So he waits and he watches the monitor. Every single time he sees the heart rate goes up, he gives the animal a reward. That is this stimulation to the brain. And pretty soon what happens is the animal starts showing more and more elevations in its heart rate. And again, every time that it does, it gets a reward. So think of heart rate then as being similar to an animal that's pressing a bar in order to get food. The only difference here is that we're dealing with this response, this physiological response, which is an involuntary response, and we are rewarding that response by administering, right, this very rewarding electrical stimulation to the brain. So he's able to do that, and he's also able to do this if he takes another animal, does the same thing, mobilizes it with this curare, and so he eliminates voluntary responses, places an electrode down into the brain of the animal, into the medial forebrain bundle. And now what he does is he's monitoring heart rate, and whenever he sees the animal show a slight decrease in heart rate, what he does is he gives the animal some stimulation to the brain. And what he finds is that over time, what he's able to do is to shape a permanent lowering of the animal's heart rate. So you can reinforce increases, you can reinforce decreases in heart rate and get sustained either elevations or sustained reductions. Now, this is an incredible experiment in the history of psychology. And again, I can't say enough about how important it is, okay, for the field of psychology. So the implications of his research, first of all, we go back to that question that I posed earlier, is it possible to operantly condition an involuntary response? You bet it is. Absolutely it is. And this provides the groundwork for this technique, which we call biofeedback, which is so important in the field of open up a whole new field of what we call behavioral medicine. And that is, treatment of individuals with various disorders by way of using learning techniques. Okay, so let's talk a little bit about this, success with biofeedback. Here you see an individual, a patient that's undergoing biofeedback training. And I'll get into the nuts and bolts of this in just a few moments. It's very much involved in terms of relaxation training. But biofeedback has been used for all these different things, for treating individuals with attention deficit disorder, to treating individuals with epilepsy, to treating people with sleep disorders, alcoholism, certain types of psychopathic disorders, asthma, irritable bowel syndrome, hypertension, migraine headaches, treatment of individuals in terms of stress and anxiety. I mean, it's been used, what we're saying is this: hey, we don't need drugs to treat these things. What we can do is we can treat these things by way of this biofeedback kind of training. Again, let's take a look at this so that you understand it. Biofeedback, the essentials of biofeedback, it's a self-regulatory technique in terms of getting people involved in controlling their own involuntary responses. Now, what kinds of response? Blood pressure, for example. Relaxation of forehead muscles that are involved in migraine, tension headaches. Reduction of extreme blushing, for example. So here you have an individual, a patient that's hooked up, measuring a variety of different physiological measures. There's a computer screen that's in front of them that is showing readouts of these various physiological measures of heart rate, brainwave activity, skin conductance, that is the temperature of the skin. The person is aware now of these ordinarily what are weak responses, weak physiological responses, but now they're enhanced. They're right in front of you, okay, on that screen, and you can see them. There's very clear signals. Sometimes they're in color, sometimes there are sounds that are associated with them. So a patient then can see their bodily reactions right in front of them, right on that screen. And again, these are signals that are being amplified. They're being transformed into lights, transformed into sounds, so that it's easy for the patient to see changes, okay, in those physiological measures. And the job of the patient is to control the level of external cues, okay, that they're being exposed to, okay, so, or that they're seeing, okay, on that screen that's in front of them. So again, let's take a little bit more. There are some very common relaxation techniques that are used in biofeedback training. Here's a few of them. Listening to binaural beats, for example, that's very relaxing for individuals. Progressive relaxation by squeezing something repeatedly. Looking at pictures of nature or of animals in nature. Deep breathing, mental imagery that is visualizing something that you want to happen to you. Thinking of every part of your body one by one. You can relieve stress by punching a bag, for example. You can relieve stress by engaging in journal writing. So these are all ways of relaxing. These are all ways of trying to modify scientific ways to modify stress, to de-stress, if you will, in a relatively short period of time. So again, examples of biofeedback training. Well, let's take the technique, a very common technique of modifying a person's skin temperature. Here's one way in which we can measure skin temperature. Through this device that we see right here, where we have these little finger gloves that are put on individual fingers, and we can take a look at skin temperature. Another way is by these that are called biodots. That again, we can measure remotely what is happening in terms of skin temperature. So you're given constant feedback then about your finger temperature. And then, a patient would be asked to raise it. And patients can see their finger temperature on a digital readout. And by way of these relaxation techniques that they learn, it's been shown, for example, that in some studies, a person can raise their finger temperature from 79 to 87 degrees. And one thing that we know is that skin warming can lead to sustained reductions in blood pressure. Okay. So again, it's making you aware of what's happening in terms of physiological responses, right? And by systematically trying to change that finger temperature, and again, you can do it by way of different kinds of relaxation techniques. That warming of your skin temperature is going to lead to a reduction in blood pressure. So again, we're saying we don't need drugs. We can do it in this way. Okay. So this becomes an incredibly important technique in treating individuals for a variety of different disorders that I noted to you in a previous slide. So let me talk about one other very important area here that a relatively new advance, I would say, over the last 30 years or so. And it all has to do with some fascinating research that was done at the University of Pennsylvania about 30 years ago, 30, 40 years ago, by a researcher by the name of Martin Seligman. So let me tell you a little bit about, there's Martin Seligman right there, a little bit about this research. If you placed a dog into a chamber like this, where periodically a sound would come over a speaker for maybe a 5-second period of time, that would signal to the animal that shortly it's going to receive a shock to the floor. But if the animal jumps over this barrier right here to the other side, and if it does so within about a 30-second period of time, then the dog's not going to get shocked. If the dog does this, comes over onto the other side, now he has a safe period of time. But again, that sound is going to come over the speaker, and it's going to be followed about 30 seconds later by a shock to the floor. But if the animal jumps back over to the other side, right, and avoids getting the shock, then we'll have a safe period. Dogs learn how to do this really well, okay? But now let's change the situation a little bit. Let's take a dog now that has an experience where it's been placed in a box, okay, prior to being placed in here, placed into a different box where the animal periodically gets shock to its paws, okay, shock on the floor. So the shock occurs at random intervals, and the shock lasts for about 5 seconds or so, and then it goes off. And the animal can do absolutely nothing to escape it or to avoid it. It's in this box, can't get out, and it's being periodically shocked. So we do this maybe for, a period of time on one day, maybe for a couple hours, where the animal's periodically getting the shock, can't avoid it, can't escape it, can't do anything about it. And now what we do is we take that animal and we place them into this avoidance box. And we try to teach this animal this avoidance response. That is, the sound comes on, short period of time, that signals that shock's coming in 30 seconds, but if the animal jumps over to the other side, the animal can avoid the shock. They never learn it. They can't learn it. They simply sit there in the corner of the cage and they take the shock. They have learned to be helpless. This indeed is a very important finding. This shows the results of the original experiment that Seligman conducted. This shows the percent of these dogs that learned this avoidance response. Here's our control animal, an animal that had no prior experience, simply placed in the box for the first time, learns the response. But the animal that received that unavoidable, inescapable shock, that has a history of it now prior to coming into this situation. Look at this animal. This animal cannot ostensibly learn this avoidance response. They have learned to be helpless. Okay. This theory, again, the animals with a history of this exposure to the inescapable shock simply fail to learn that avoidance response. Instead, they learn to be helpless. This now is probably the leading theory to explain depression. So when we look now at the implications of this learned helplessness, Martin Seligman says, there are people in the world, sadly, who have experiences that lead to learned helplessness. They're involved in situations where repeatedly, for one reason or another, they simply have no success whatsoever in terms of, very difficult situations that they're in. They become depressed. And indeed, their motivation goes way, way down. Right? And indeed, their expectations, because of this helplessness, because of these situations that they've been in, which they haven't been able to properly successfully encounter, their expectations for themselves become lower. What Seligman also says is this has all the characteristics of a personality trait. People who become optimists in their lives because of the fact that they have success with difficult situations, and those who become pessimists, who are involved in situations repeatedly where they have no success. So indeed, he's done a lot of research, for example, to show that individuals with personality traits of being an optimist, if they're diagnosed with cancer, are much more successful, for example, in terms of beating the cancer, as opposed to individuals that are pessimists. It has a number of different implications, okay, for how we conduct our lives. So if you have experiences with insolvable tasks, those experiences have dramatic carryover effects for you in the future, right? And indeed, we simply cannot minimize this in terms of it being a very important theory of how different types of behavior disorders may in fact be learned, that individuals learn to be helpless. So this has produced another area, rapidly growing area in psychology, which is called positive psychology. It's ostensibly the study of happiness. What makes us happy? Indeed, psychology traditionally has been focused on dysfunction. It's focused on mental illness. It's focused on psychological problems and how to treat them. That's not irrelevant. That's fine. That's all fine and good. But what positive psychology is saying is, hey, there's a whole other way of exploring this, of exploring human behavior, and that is what makes us happy, what leads to happiness. So it's really produced a movement almost worldwide in terms of studying, again, positive psychology. So positive psychology is a new field. It's examining how ordinary people become happier and more fulfilled, and it is now a very, very important part of psychology. Go back and take a look at the development of all this. It really required us to understand learning. It really has required us to understand some of those basic principles of learning that we talked about in terms of classical conditioning and operant conditioning. So this really brings to a close our analysis of learning. We're going to be moving on to the area of vision. But indeed, this is, a topic that is so important, the study of learning, for so many different aspects of the study of behavior, the study of psychology. So in our next lecture, we'll be taking a look at vision.